So the transcription in eukaryotes is undertaken by polymerases. They are closely related to the RNA polymerases found in the prokaryotes. The process of transcription is identical in both the prokaryotes and eukaryotes. There are, however, differences in the machinery used in each case. So the process is actually the process of formation of mRNA from DNA is same in eukaryotes and prokaryotes, but the machinery involved, the factors and the enzymes involved, they are quite different. Bacteria have only one RNA polymerase, but all eukaryotes have at least three different ones. That is RNA polymerase 1, RNA polymerase 2, and RNA polymerase 3. And plants also have polymerase 4 and polymerase 5. So there are 5 different types of polymerases found in the animals and plants. In addition, whereas bacteria require only one additional initiation factor, and that was sigma factor, several initiation factors are required for efficient and promoter specific initiation of transcription in eukaryotes. These are called general transcription factors or GTFs. In vitro, the general transcription factors are all that are required together with POL2 to initiate transcription on a DNA template. So in vitro, that is out of the cell, if we carry out this transcription, all the general transcription factors plus DNA RNA polymerase 2 are required for the initiation of polymerization. In vivo, however, the general transcription factors are not alone sufficient to bind promoter sequences and elicit significant expression. So in vivo, there are some things more required. Rather, additional factors are required, including DNA binding regulatory proteins, the so-called mediator complex, and often chromatin modifying enzymes, because in eukaryotes the DNA is in the form of a chromatin. The eukaryotic core promoter refers to the minimal set of sequences or sequence elements that are required for accurate transcription initiation by the POL2 machinery. A core promoter is typically about 40 to 60 nucleotide long, extended either upstream or downstream from the transcription start site. So on the DNA, there is some transcription start site, and this core promoter, which is about 40 to 60 nucleotide long, it is present either on the upstream or downstream side of this start point. The elements found in POL2 core promoter include the TF2B recognition element, that is BRE, the Tata element or Tata box, the initiator INR, and the downstream promoter elements known as DPE, DCE, and MTE. So these elements are present in the core promoter. Here you can see this is a representation of the core promoter. So you can see this is BRE, this is Tata box, and this is INR, and then this is DPE, and so there are many such elements which are present in the POL2 core promoter. Typically, a promoter includes some subset of these elements. This subset may be different in different organisms. Thus, for example, promoters typically have either a Tata element or a DPE element, not both. So in one promoter, this core promoter, either TATA element is present or DPE is present. Often a TATA containing promoter also contains a DCE. The INR initiator is the most common element found in combination with both TATA and DPEs. So this INR or initiator is the most common element which found in combination with TATA and DPEs. Typically, upstream of the core promoter, there are other sequence elements required for accurate and efficient transcription in vivo. So in vivo, there are some additional elements which are present upstream of the core element, core promoter. Together, these elements constitute the regulatory sequences. So these regulatory sequences are also required for accurate and efficient transcription. These elements include promoter proximal element, upstream activator sequences or UAS, enhancers, and a series of other elements called silencers, boundary elements, and insulators. So all these regulatory elements are also required for the initiation of transcription. All of these DNA elements bind regulatory proteins such as activators and repressors, which help or hinder transcription from the core promoter. So the transcription will start from the core promoter, and then these activators or repressors will bind with this complex. And if activators bind, transcription will take place, and if repressors will bind, they will hinder the transcription. Some of these regulatory sequences can be located many tens or even hundreds of kilobases from the core promoter on which they act. So their location is very wide, maybe tens or even hundreds kilobases away from the transcription site.